A very good morning, everyone. Thank you for coming to the machine learning for brain imaging session. So today, together with all my colleagues, Neil Oxtoby, Tim Wang, as well as Vince Cajon, who just went to the speaker room, and as well as Danny Alexandra, who couldn't be here today, we jointly organized this session. Hopefully, we could promote some interactions as well as communications and collaborations across the OHBM community to promote machine learning for brain imaging so that we can learn together. So I'm Helen Zhou from National University of Singapore, and without further ado, let's start. So the first speaker today is Associate Professor Neil Oxtoby, and who is from University College London. And he's really an expert in machine learning for brain imaging. And today he's going to tell us data-driven modeling for clinical trials on Alzheimer's disease.
Good morning. How are we? The middle day of the conference. My name is Neil Oxtoby. I work at UCL. But you may be able to tell that I have a more local accent. I did my high school and undergrad and PhD in Brisbane, so it feels like home. So I'd also like to acknowledge the traditional custodians of this land. They did a fabulous job looking after Brisbane, making it awesome, keeping it awesome for 10,000 years. Long may that continue. So I'm going to tell you about what we do in the Progression of Neurodegenerative Disease Group, POND, at UCL, and particularly a specific application of a disease progression model. I am available for consultancy, so I can make my disclosures slide longer if you like. I do consult for all the topics that are in my talk today, but it's unrelated to industry money. This is funded by a research grant at the university. Starting with acknowledgements, we like to do things backwards. Most of the work that I'm going to talk about today in the case study was done by postdoc Cameron Shand, who's moved on, and Ann Zhao, but also key people in this work are Danny Alexander, Alex Young, and Gonzalo Castro. Lots of collaborators. We work with clinicians, neurologists, and machine learning folk. So, broadly, 3 things will be touched on today. Data-driven disease progression modeling, and specifically an unsupervised learning flavor of that with some clustering. Obviously, brain imaging is heavily involved. That's why I'm here. And I'll talk about clinical trials on Alzheimer's disease, and specifically about how machine learning can help with trial enrichment, and a little bit about some of these new drugs that are coming out, obviously, and a case study on an actual trial, some actual patient-level data from a phase 3 clinical trial. And I'll tell you some results in applying my models to that. My motivation for this and my current line of work, heavily applied work, was that I was reading all these machine learning papers, specifically in Alzheimer's disease, and they all make bold claims. The discussion and conclusion section says, we're going to revolutionize clinical trials, we're going to change healthcare. And then they write another paper a couple of months later, a couple of years later, and they haven't done anything about it. Then I realized I was saying the same things in my papers. So I thought I'd put my money where my mouth is, which is difficult in clinical trials in Alzheimer's disease, specifically because the drugs haven't been working for so long. The trials have been so-called failing. So the pharmaceutical companies that sponsor these trials don't like to release the data. All you see is the average in a plot on a paper in New England Journal of Medicine. You don't get to see the patient-level data, and particularly the brain scans, until quite recently. So I've been on a quest for supermodels and drugs, where I build these computational models and I try to understand how the treatment response varies. So let's get into it. Data-driven disease progression modeling. It's a machine learning method, a mathematical modeling method that both learns the disease timeline on the horizontal axis at the same time as learning the pattern of disease progression, the severity on the vertical axis. So those who work in Alzheimer's disease will be familiar with this sigmoidal pattern. It's where disease trajectories in different biomarkers become abnormal, vertical axis, in a sequence. So there's one first, second, third. And we can reconstruct these timelines using our data-driven disease progression modeling methods, sometimes from even cross-sectional data. So we get these long time trajectories from just short time, couple of years, snapshots of biomarker measurements, including brain imaging, from multiple individuals. For more details, you can go check out Alex Young and my review paper from last year. A specific one of those that was invented at UCL is the subtype and stage inference, or SUSTAIN algorithm, where you take these heterogeneous patient snapshots of different levels of severity. There's this color map here for an individual brain scan from each patient. And the model both learns the sequence, the progression, longitudinal progression sequence, and clusters, so subtypes of disease progression simultaneously. It's a flavor of unsupervised machine learning. And what we get out of it is these disease progression subtypes from cross-sectional data even. That was invented by Alex Young and Danny Alexander, and you can read the original paper in Nature Communications 2018. A particular application of this from Jake Vogel and colleagues was in tau PET. So tau is one of the primary pathologies. It's a misfolded protein in the brain. You can measure it in vivo. And Jake applied tau PET on a large study, so almost 2,000 patients, and found 4 subtypes of tau accumulation across the brain. They accumulate in different regions at different times. And some useful differences between those subtypes, so different cognitive deficits, neuropsychological deficits, biological differences in genetics, different age. So this is potentially a useful stratification of people into disease progression subtypes. I've done some talks on this in various conferences and things, so you can check those out at my website. And if you're interested in tau spreading in general, there's a poster that's been presented yesterday and today by Andre Altman. You may have seen us wearing these t-shirts around. He'll tell you more about tau spreading in Alzheimer's disease. So that's disease progression modeling, something on clinical trials now in Alzheimer's disease. There's lots of cool imaging that we can do in Alzheimer's. So these are 3 different types of scans, left to right, normal in the bottom and abnormal at the top. On the left here, we've got an amyloid PET scan, the primary pathology in Alzheimer's disease. So you get the brain goes hot in amyloid plaques. In the middle, tau PET. It's a bit more localized spatially. But again, you get an increasing heat map. And FDG is a measure of glucose hypometabolism, where you have deficiency, so hypometabolism, the heat map goes colder. So you can use these biomarkers to understand the disease progression. And after doing a lot of these measurements in different parts of the world, this culminated in a hypothetical model of the sequence of abnormality in Alzheimer's disease as a sequence of dynamic biomarker changes that happen one after the other in this sort of stereotype manner. So that's Cliff Jack's work. So you can use that information in a clinical trial to help find the right people at the right time. You can start with basic screening of people who are at risk for a disease. In Alzheimer's, the biggest risk factor is age. And you could do some very basic clinical measures at this point to make sure that someone might be eligible for a trial. Then you look at some exclusion criteria, someone who's got some co-pathology that might confound your analysis in your clinical trial, so you rule some people out. Structural MRI is often involved there. And then you can screen in individuals, again, with higher risk. Typically, biomarkers are used there, including imaging, PET imaging, CSF, cerebrospinal fluid measures of protein. And you can use more imaging, again, to enrich for the risk of decline. And all the way along, the number of subjects that could potentially be enrolled in your trial decreases. And the cost goes up. But the idea is to find the right people at the right time. These 2 latter stages are what's called enrichment of a clinical trial. The basic idea behind enrichment is to get the right people at the right time. So potentially treatment responders, people at risk for the disease, who are about to decline with some outcome measure for Alzheimer's disease. That's typically a memory test of some form or another. So we can use machine learning to help enrich for that. The good thing about enrichment is that you end up with a smaller trial population. So if your new drug that you're experimenting with has some side effects, you can minimize the exposure to that. Save some time, obviously, save some money, fewer brain scans, etc. And in the spirit of personalized medicine, you can find the right people. It does limit the prescription, so you need to just keep it real about this. So if you've done all these measurements to find the right people at the right time, then you have to do the same ones again when you give the drug out, when you prescribe it. Worth keeping in mind. So if I was to give a course on this, Clinical Trial Enrichment 101, the aim is to increase the statistical power of your trial. And that can be decreasing the heterogeneity in your cohort, finding high-risk patients and people who are likely to respond. In Alzheimer's disease, that's amyloid-positive individuals who have this pathology in the brain, preferably with no vascular pathology, a confounding risk. Someone who's about to decline, someone with mild cognitive impairment, for example, a typical prodrome of Alzheimer's disease. And patients who might be likely to respond to a drug, for example, if they are negative for a genetic risk. We don't understand the disease well to be able to do that too much. And these are called practical, prognostic, and predictive enrichment. So, onto application. Okay, got time. Good, good. The A4 study, asymptomatic Alzheimer's disease. It was a phase 3 clinical trial. I'm going to talk about the SUSTAIN model that I pre-trained on data from an observational study. So I have this disease progression subtypes model, and I used it, deployed it, tested it in the A4 trial data, and then did a post hoc subgroup analysis. So the trial's finished. I just want to see if this subtype stratification based on my machine learning model could potentially be useful, give us something that could potentially be used to enrich a trial in the future. For the SUSTAIN model, the input was T1 weighted structural MRI, just brain volumes from the Alzheimer's Disease Neuroimaging Initiative, ADNI dataset. We processed this with the Free Surface software suite from Boston into regional brain volumes across the neocortex and the subcortical regions. And then converted those brain volumes into Z-scores, normative scores relative to a control population who are all amyloid negative and cognitively normal. So these are people who don't have biological Alzheimer's disease, have no symptoms, no cognitive decline. This requirement resulted in 1,438 individuals across the diagnostic categories available in the ADNI dataset, about 100 cognitively normal individuals, nearly 1,000 with mild cognitive impairment, including individuals who did and didn't later on progress to a diagnosis of Alzheimer's dementia, and 360 with an Alzheimer's diagnosis. They were all amyloid positive, either determined on an amyloid PET scan of the brain or through the protein measures in a cerebrospinal fluid, CSF measure. I used the PySUSTAIN software, translated from MATLAB by Leon Axman and Pete Wijeratny, which you can see on it's openly available on our group's GitHub page. Plenty of tutorials and videos and things. So I plugged in some brain volumes into this clustering disease progression model, and this is what I got. So severity is increasing from yellow to purple. This top line, I've called it a limbic subtype. It looks like the typical Alzheimer's disease type pattern. There's early atrophy in the hippocampus and amygdala, proceeds to the temporal lobe, and then beyond. They all end up at the end converging on full severity across the brain. The thing that distinguishes them is the early changes in the combinations. So the middle subtype here is a cortical subtype where the hippocampus wasn't affected until a little bit later. The early stages were the neocortex. And then a subcortical subtype, which we're still trying to understand, where putamen, pallidum, some of these deep grey matter structures were affected early. So these Z-scores take us into the tail of a normal distribution, a Gaussian distribution. Z equals 2 is about the 98th percentile. You can visualize this slightly differently. Oh, there's also a fourth subgroup, stage 0. These individuals had no atrophy on this scale here, this Z-score scale, so they basically looked like the cognitively normal amyloid negatives. They had very healthy looking brain. 14 seconds. This will be a challenge. Here's a different visualization, going left to right. It's like a top-down look of one of those sigmoids, which is another way to look at it here. In the limbic subtype, the hippocampus was early. The cortical subtype, it was somewhere in the middle. And the subcortical, it was later on. All have the hippocampus affected, which is the memory center of the brain. All seem to be subtypes of Alzheimer's disease. But what do they look like? So the limbic subtype, as you might expect, had more Alzheimer's diagnoses and more cognitively normal amyloid positives versus the stage 0, which was the opposite of that. Makes sense. They had a higher proportion of ApoE4 carriers, which is a genetic risk factor for Alzheimer's disease. And again, stage 0 subgroup with no atrophy had the opposite of that, lower count. The subcortical subtype had more males. And then basically the limbic typical Alzheimer's disease subtype had all the biomarker evidence of being typical Alzheimer's disease versus most of the other subtypes. So there's this A4 trial. They trialed this drug called solanezumab, which attacks soluble amyloid floating around in the brain. The result for the trial was negative ultimately. This is the primary outcome, the PAC, preclinical Alzheimer cognitive composite. It's again a Z-score based thing. Worsening is a lower score, negative score. The placebo, you can see in blue, actually did better than the active drug arm, not statistically significantly. And this is 240 weeks was the primary readout, about 4.5 years. It's a very interesting spline model for the stats geeks. Go check it out. So when I stratify by my subtype model, you can see that there's differential response. So same outcome, same vertical scales, a Z-score worsening to the bottom, and the different colors depict the subgroups that I mentioned, the normal stage 0 looking group and the 3 subtypes of Alzheimer atrophy. Now, these are the proportions here. You can see the subcortical one's quite small. And in this particular trial, this preclinical amyloid positive individuals, their cortical subtype was the dominant subtype, but most of them had pretty normal looking brains. As you'd expect, they have no cognitive dysfunction. So this is on the left, this is the placebo arm split into the subgroups, and on the right is the active arm, solanezumab, split into the subgroups. If we look within subgroup, on the left here we've got the typical Alzheimer subtype, severe decline, this is a Z-score of -8 on the preclinical Alzheimer composite, but again, the placebo did better in blue. On the right, you've got the subcortical subtype, which bucked this trend, which is quite cool. The drug didn't work ultimately, but for this subgroup, it started to. Quite nifty, quite cool. Again, pretty small numbers, 3% of 1,100 individuals. But it shows that the model can be useful and may be useful for other drugs. So I encourage the pharmaceutical companies involved in all clinical trials in Alzheimer's disease to release that patient-level data if they could. So here's the summary. We did an atrophy model, basically, using machine learning on brain imaging, the SUSTAIN model, which showed both predictive enrichment capability, being able to predict who's going to decline. No surprises, this was the limbic typical Alzheimer's subtype. And prognostic enrichment capability as well, who might respond. The subcortical subtype seems they will respond. The bad news is it's only 3% of the individuals, so not many people coming into a memory clinic would be eligible for the drug if the decision for the clinical trial outcome was made based on these subgroups. Thank you very much.
Thank you, Neil.
We might have a burning
Yeah,
question.
we have a burning question, so please.
Thank you. Can you tell us a little bit more about the unsupervised learning method that you used?
Yep, sure. It's called SUSTAIN. It works on cross-sectional data, and it's an event-based model. So if I was to see someone with, say, a cold or flu, and if you can assume that symptoms always only get worse, I would see some patients who have a cough only, some who have a sneeze only, and some who have both. The clustering algorithm would work that out just by plugging the data in, and more symptoms than just cough or sneeze, and it would pull them into subtypes, a cough first and a sneeze first. And then that analogy carries on to atrophy.
In case of brain images, what's the input? Is it the whole image?
Brain volumes, regional volumes from the FreeSurfer Desiccant Kiliani Atlas.
Easy. Thank you.
So we have another burning question. After that, we have to move on, but you can still
we'll
ask.
do questions at the end, so please do stick around.
You can
yeah.
still, you know,
you
can use the app to enter the questions, and we will answer all questions at
We'll
the
quickly
end.
do this one, though, yeah.
Please.
Sorry. Hi, very good talk. I was wondering if you encountered any problem with FreeSurfer when you input the data with patient with Alzheimer's. Sometimes FreeSurfer will not be able to give you the kind of normal sort of output.
Yeah, absolutely.
And how would you deal with that? Because now you're dealing with the data that a little bit different from the normative samples.
Yeah, so most of the FreeSurfer errors were the Talarac stage, the registration to M&I space, and you can fix those manually. Most of those there was no bias towards that happening more in the Alzheimer's patients with the more severe atrophy, which is quite interesting. FreeSurfer 7 is quite robust. Thank you. Look forward to more questions later.
Yeah.